Okay, now I'm starting. Okay, like I said, I'm gonna try and keep this short. I am going to give you a very simplified snippet from a bigger story, and if this piques your interest, then these are our publications. The details are here. Okay, so I'm going to start with this famous figure. I'm sure you've seen this before. Many of you have seen this before. This is the epigenetic landscape that was presented by Conrad Waddington back in the wait. Oh, my mic was off. You're fine. Now my mic is on. Okay, now you can hear me. So this is the famous epigenetic landscape that was presented by Conrad Waddington back in the 1950s. And back then, we have to remember that molecular biology and cell biology was not really a thing. So this is just an imaginary, very metaphorical landscape. I feel like people cannot see me. And so Conrad Waddington imagined this cell, I guess a stem cell, and it's dividing, and the cells start rolling down this mountain, this mysterious mountain, and some of the cells go this way and become your muscle cells, and other cells go that way and become your brain cells or blood cells, and so on and so forth. And today we call this process, this magical process, cell differentiation. And today we also have a better idea of some of the molecular mechanisms that form the basis of this magical landscape. And we call those mechanisms.
You can't be well.
You cannot. Oh.
So it's just.
Can you hear me now?
Probably better.
Is it better? Yeah. Okay, so I'm going to stay close to the mic because I keep walking away from the mic because I'm a lot more dynamic. Anyway, so all right, so we call these mechanisms epigenetic mechanisms, and I'm going to just talk about one, and that is DNA methylation. So many consider this to be the most canonical of the epigenetic mechanisms. It's the closest to the DNA. It involves a direct chemical modification to the DNA sequence. So a cytosine gains a methyl group. So a CpG becomes a methyl CpG. Now, this is another way to visualize the epigenetic landscape, but this is specifically the DNA methylation landscape. So what we did here was we measured the methylation level in thousands of CpGs that are all over the mouse genome. And this is simply the histogram showing the frequency distribution of the methylation levels. And this looks a lot different from what you see in RNA-seq and proteomic data because DNA methylation has a very distinct bimodality. What that means is the DNA methylation, depending on where the CpG is located, is relatively predictable. The x-axis here is the beta value, which is basically just the fraction methylated. So 0 means totally unmethylated, 1 means fully methylated, 0.5 means 50% methylated. So many of the CpGs in the mammalian genome tend to have low methylation. So they're closer to 0. Most of the CpGs tend to be highly methylated, closer to 1. And then there's a handful of CpGs that are kind of indecisive and are somewhere here. And that means that the mammalian epigenome is highly informative. It's predictable, it's well-ordered and informative. Now, this is the same plot, but I'm just showing that for two mice. It's a BXD animal. One is old, over 2 years old, and the other one is young, about 6 months old. And you can see that this is very subtle, but you can see this shift in the landscape. So in the old mouse, the red plot here, the hypo and hypermethylated peaks have kind of eroded with time, sort of a landscape erosion. And there's a slight shift towards a more random state. This very subtle change in the distribution pattern can be used to compute the overall information content in your DNA, which we call epigenetic entropy. So entropy basically measures the level of disorder that you have. And we and others have shown that this entropy, this epigenetic entropy, increases with aging. And that's true whether you're a mouse or you're a human, and probably whether you're some other mammal. And this is just the epigenetic entropy in the BXD mice that are 6 months old and older, 21 months old. Okay. Now, to give you an idea of one of the CPGs that might be contributing to this shift in the landscape, this is one example CPG. The CPG is located near the promoter region of a gene called JARET2. This is a polycomb repressor gene. It's very important in early development. And these are about 340 BXD mice. We measure DNA methylation, and they have like they range in age. And you can see that when the mice are kind of young, the methylation at this CPG is closer to 0.2. And as the mice get older, it slowly shifts towards 0.4. So then the question is, does this simply represent a random process, like a random accumulation of error? Because when people think about entropy, they're thinking about randomness, right? And or is it a regulated process? So there's this idea that the increase in randomness in complex biological systems is inevitable because it's simply following the arrow of time, the second law of thermodynamics. But what makes an individual resilient to that shift could be a genetic trait. So to examine that, we ask this question. After we control the effect of aging, do the CPGs that change with age, means the CPGs that gain methylation with age or lose methylation with age, are they also genetically regulated? And for that, we performed methylation QTL mapping, very similar to protein QTL mapping, but we call them MEQTL. And this is the MEQTL plot for the JARET2 CPG. And this triangle here tells you where that CPG is located, physically located in the genome, in the mouse genome. That's on chromosome 13. But the genetic region that controls its methylation is on chromosome 5. So what this tells you is that the variation that you see in that CPG is partly due to aging and partly due to genetics. Now, this is the same plot that I showed you earlier from the 340 animals, but now I've color-coded each animal by the genotype in that locus, in this chromosome 5 locus. So these are BXDs, so they get either a B allele or a D allele. So you can see that the animals that inherit the D allele in the MEQTL tend to have a higher methylation. So since this CPG gains methylation with aging, if we say that a higher methylation means higher biological age, then the animals that have the D genotype here are epigenetically older. So they have a more aged condition. So this looks blurred. Okay. So JARET2 is just one of the CPGs that we looked at. We had measured thousands of CPGs. So we performed the MEQTL mapping for the thousands of CPGs. And this genome graph is a very simplified representation of the main result. You've seen this genome graph before. The y-axis tells you where the CPG is located. The x-axis tells you where the MEQTL is located. And right now, I just want you to focus on this trans band on chromosome 5, because that tells you that that chromosome 5 region is a regulatory hotspot that is controlling the methylation level of many CPGs that's located all over the mouse genome. And the interesting thing is that many of the CPGs that map to the chromosome 5 locus tend to gain methylation with aging. The other thing is that many of the CPGs are located in genes that are involved in early development, just like the JARET2 gene. And having a D genotype at this locus results in a higher methylation of the target gene. Now, this locus is really gene dense, and there's a lot of really good candidates. But right now, my favorite candidate gene that's right here is a transcription factor called HNF1A. It's a hepatic nuclear factor. And I wouldn't go into details as to why I think that's my favorite. But anyway, this is my last slide. So that observation has led us to a model or a hypothesis on how your genetics could influence the epigenetic landscape that's formed during early development and how this can impact your aging trajectory. So the model is you have a genetic variant, right? You have chromosome 5, there's this MEQTL that comes in a B allele or a D allele. And this chromosome region, genetic region, produces a certain factor. We're not sure what that factor is, but right now my favorite possible candidate is a transcription factor. And one of the reasons why this is my favorite is because many of the CPGs that are transmodulated by this QTL happens to be in downstream targets of HNF1A. And many of the CPGs are located in sequences that have binding sites, for the hepatic nuclear factors. So it produces this factor, and because of the genetic variation, you have there's a D variant and a B variant. And because of this variance, in the protein, also HNF1A has a missense mutation in the BXDs. So because of this variation, it could possibly be resulting in differential methylation of the downstream CPGs during development, right? So in the B, you have lower methylation. In the D, you have much higher methylation. And since these very sites are the ones that increase in methylation with aging, not just in mice, we've seen this in mammals, like in humans as well. Many of the target regions do gain methylation, because the D starts out with a much higher methylation to begin with. As they get older, their methylome appears more aged. And this could have a downstream impact. And we've seen a small evidence that this could be linked to longevity and potentially body weight. So this is the tiny story. And so why do we care about this? We care about this because I think that this is telling us something really interesting about the fundamental links between early development and aging, what people call the developmental origins of aging. The HNFs are just really interesting. It is linked to many metabolic genes. So one of the targets of HNF is the insulin growth factor 1. So if you knock out HNF1A, you're both diabetic and you have dwarfism. And so this is like an interesting point where there's this intersection between development, body size, metabolism, and aging. That's it. And these are the people I like to thank.